sticking with me until the end. So I'm working with Malar as well in leveraging preclinical MRI from a slightly different angle, I would say, from what you've seen so far in the previous presentations. First of all, because I'm working on fMRI rather than structural analysis, but also because I'm working more on the development of better analysis strategies, and I'm trying to leverage tools that are available in animals and not so easily available in humans to then inform how we can optimize our analysis strategies more broadly across the field. And this is what this presentation is going to be about, more specifically with our technique I call neural prior recovery to analyze brain network using resting state fMRI. So I will begin with a cartoon that summarizes quickly the complexity of separating the different signal contributions in fMRI. So as many of you must be aware, fMRI is a powerful tool for investigating brain function, given its access to brain-wide activity. However, it comes at a cost of complexity in disentangling the different contributions to the signal, since non-neural sources, such as respiration or motion, can interfere with analysis and cause spurious results. For instance, in that paper, they show how without accounting for motion, you don't recover the full extent of the default mode network. And that's why it is common to try to model these non-neural contributions to the signal and remove them before analysis. But in practice, it is very complicated to do so in a reliable fashion while removing the full extent of these potential confounds. And that's why we still rely nowadays with resting state analysis with less specific techniques, such as the regression of the global signal itself, which more effectively remove the full extent of confounds, but comes at the cost of also removing some neural signal of interest. And of course, this is a gross summary of a very complex topic here, but here I'm just highlighting that we still lack a standardized, reliable approach to specifically look at neural sources in resting state connectivity analysis. And this is arguably one of the contributors to ongoing reproducibility challenges in the field. And now where I'm going to take you in this presentation is through a different angle to try to tackle this problem, where I'm arguing that if we define a prior for what we expect to see as neural activity in the scan, we can mitigate some of these issues. But now first, what do I mean by a neural prior? Where in this case, with connectivity analysis, I'm referring to spatial maps that represent canonical networks, which have been described in the human literature for multiple decades now, and are very reproducible at the group level across many different studies, and they remain the main ground for the interpretation of connectivity results in the literature. And so they inform really what we expect to see in an individual scan when we're doing individual level analysis. However, it is challenging in humans to really look at the neural underpinning of these networks, and that's why I'm working with mouse fMRI to combine the technique with more invasive tools, more specifically here, cortex-wide calcium imaging, which offers a more direct assay of brain-wide activity. And with this technique, we can carry parallel analyses in each modality to relate signatures of brain networks seen with fMRI with a corresponding pattern of activity in the calcium modality. In this case, I'm using group IC decomposition to identify brain networks that more specifically I'm showing the somatomotor mouse network found in both modality, and that's what I'm going to refer to as a neural prior in this presentation. Now, after having defined what is the particular prior of interest that we want to look at, the challenge is to isolate this particular source of signal from the set of potential confound. And that's the goal of the technique I'm going to present, called neural prior recovery, or NPR, which takes as input a set of spatial map representing networks of interest, and will find all of the sources which are not this prior in a particular scan, so that we find all potential confounds and remove them. And this is done using complementary PCA, which I'm going to describe in the next few slides. This is building on a pre-existing connectivity technique called dual regression, which much before us suggested that by starting with a group IC decomposition of the entire data set to find the predominant sources of signal that are found across the data sets corresponding to either different brain networks, but also certain confounds. They suggested that we can use these as prior for what we want to recover in the individual scan. And with dual regression, this is done by first regressing these components to obtain time courses that are specific to this scan for each component, and then regressing the time courses to obtain then their spatial counterpart, now obtaining scan-specific version of these networks. However, this was not necessarily designed in line with state-of-the-art recommendations for confound correction. More specifically, the group components do not describe scan-specific manifestation of confounds, and this is known to be an important limitation, which we wanted to mitigate. And so building on top of dual regression, we were able to exploit a particular property of the technique whereby providing the outputs of one iteration of dual regression as the input to another iteration, this iterative process converges on a set of spatial map that maximize variance explained in a target scan. And this is actually equivalent to PCA, principal component analysis, which is a technique that will find the particular sources of signal found in a specific scan. And the reason why this iterative formulation of PCA is useful is because you can modify it to include a set of priors at each iteration, keeping these fixed maps as fixed maps across iterations and enforcing the other components to differ from this set of prior, because that part of the variance is already explained. Thus, we're finding a set of scan-specific components which are not the prior, which I call complementary PCA. And so coming back to this schema here, this is achieving the goal that I mentioned, where we want to find all of the scan-specific set of components which can be confounds, but are not the prior network of interest. And to fully complete the NPR process, there's a second step where now we're providing CPCA as input, the set of confound components, such that if we accounted for all sources which are not the prior, the only source that will remain to be fitted is the actual network of interest that we wanted to analyze, but now providing a scan-specific version of this network. Now, on this slide, I'm quickly covering how this process can converge, providing an example in one scan. On the left, I'm showing how the final output changes depending on the number of confound components fitted, and eventually you obtain you do recover the network of interest. I'm showing in the middle the same thing, where on the y-axis you have the correlation between the output and the prior component, and you can see eventually it plateaus. And a second criteria we're using to automatically choose the ideal number of components for a specific scan is going to be the difference between the current output and the one from the previous iteration. And so in that case, once this is minimal for a sufficient number of iterations, we can assess convergence. And so we can automatize what's the ideal number of components for a specific scan to avoid overfitting. And also I want to mention that this process can be easily modified to include multiple priors. So this is not limited to only looking at one network at a time. Now, however, even with these innovations, this was not enough to correct for the full extent of potential confounds with fMRI. More specifically, global signal artifacts were not corrected still. I'm showing on the left the Carpet plot for one scan, where you can see the signal fluctuations for all voxels across the brain, and the white and black stripes represent fluctuations in global signal. At the bottom, I'm showing the time courses for different confound components obtained with dual regression, showing that these do capture the global signal effect. But also at the bottom are time courses for the somatomotor network with different connectivity analyses, and these were all affected by global signal, including NPR. So the final innovation that was necessary was to add another CPCA step to NPR, where now we're fitting also temporal principal components, which were able to effectively mitigate these global signal effects. Now, the technique was systematically tested across 12 different mass fMRI acquisition sites, covering a range of data quality. The first measure we looked at was the ability to recover the network of interest, more specifically, the spatial correlation between the scan-specific network map and the prior canonical network map. And you can see that seed-based connectivity achieves lower correlation than dual regression or NPR. And the other metric we looked at was the correlation with confound, more specifically the temporal correlation between the network time course and the set of confound time courses obtained with dual regression. And in this case, NPR achieves lower correlation than dual regression, thus finding the best trade-off between less confound effects, but also decent network detectability. Now, just to demonstrate the effect of global signal regression here, we can see that applying this before carrying out analysis reduces network detectability, in particular for dual regression, showing how it can remove signal of interest. But the main results I want to highlight on this slide is that if no prior confound correction was applied before analysis, NPR can achieve the lowest overall confound correlation while preserving network detectability, showing how the technique is sufficient on its own to automatize the confound correction process. I'm going to end with a final set of very unique analysis that I was lucky to carry out thanks to a collaboration with Evelyn Lake at Yale University, who has acquired a unique data set with simultaneous cortex-wide calcium imaging and fMRI, thus allowing me to compare how different analyses can capture underlying network neural signature. And this is what I did by carrying out parallel analyses in each modality, by first deriving group IC components for the somato network in each modality, then deriving network time courses, and then cross-correlating these time courses after the convolution of the calcium time course for hemodynamic response. And I'm showing on the right the results where NPR achieves on average the highest correlation, and that remains true even without extra confound correction applied. So this is showing how NPR can maximally preserve the neural signature of interest. So I believe overall in this presentation, I've shown how this new framework can achieve reliable confound correction automatically while preserving specificity for neural activity. And I believe there's potential for generalizability as well, since this technique really only requires one key decision step, which is the number of components fitted, and this can be easily automatized as I've covered. But you might have been thinking about the main trade-off here, which is that, of course, our analysis is constrained to a set of priors, and we're discarding residuals. So this is not appropriate for more exploratory studies. But I would argue that for an important set of the literature, we are still relying on a canonical network for interpretability, and the rest of the signal cannot be easily validated. And so I think this conservative framework might help tackle some of the ongoing reproducibility challenges to carry out more reliable network analysis. So with that, I'll thank you for your attention. The code for the NPR implementation can be found as part of the online preclinical fMRI pipeline that I've developed. And don't hesitate to reach out to me if you have any question. And I'll end with thanking my supervisor, Malar, my collaborators, my lab, and funding sources.